Gene expression is the blueprint of cellular function, but traditional methods destroy the map. Bulk ribonucleic acid sequencing averages millions of cells, completely masking critical biological variation and tissue heterogeneity. Single-cell ribonucleic acid sequencing identifies distinct cell types but requires tissue dissociation, which destroys the native spatial context. Tissues, such as the tumor microenvironment or the brain cortex, are highly organized structures where cellular function is dictated by precise location and intercellular interaction. Spatial transcriptomics was developed specifically to overcome this fundamental limitation. Spatial transcriptomics measures the entire gene activity profile while preserving the original coordinates within the intact tissue section. This spatial information is essential for understanding complex biological processes like cell differentiation, disease progression, and the intricate mechanisms of intercellular communication. Spatial transcriptomics methods measure the entire transcriptome or a targeted subset of transcripts, linking each molecular measurement to a specific physical location within the tissue. These technologies are broadly classified into two primary categories: sequencing-based and imaging-based approaches. Sequencing-based methods, exemplified by the 10x Genomics Visium platform, capture messenger RNA onto spatially barcoded arrays. These methods offer comprehensive whole transcriptome coverage but typically operate at lower resolution, often using capture spots 50 to 100 micrometers in diameter. Conversely, imaging-based methods, including multiplexed error-robust fluorescence in situ hybridization and sequential fluorescence in situ hybridization, utilize sequential hybridization and fluorescence microscopy. These techniques visualize and identify individual transcripts directly in situ, achieving true single-molecule resolution, but they are generally limited to targeting a predefined panel of genes. The 10x Genomics Visium platform is the most widely adopted sequencing-based spatial transcriptomics method. It utilizes specialized glass slides coated with millions of oligonucleotide probes arranged into defined capture spots, typically 55 micrometers in diameter. Each capture spot contains probes featuring three essential functional elements. First, a polydoxythymidine sequence facilitates messenger RNA capture. Second, a unique spatial barcode identifies the spot's precise coordinates on the slide. Third, a unique molecular identifier tags each individual messenger RNA molecule. A thin tissue section is placed directly onto the slide and then permeabilized to release the messenger RNA. The released messenger RNA binds to the underlying probes. Reverse transcription generates complementary deoxyribonucleic acid, which incorporates the spatial barcode. Subsequent next-generation sequencing and computational alignment map the resulting gene expression profile back to the precise spot location on the corresponding tissue image. Achieving true single-cell resolution requires moving significantly beyond the 55 micrometer spot size of standard Visium. Newer methods like slide-seq and high-definition spatial transcriptomics utilize barcoded microparticles known as beads to dramatically increase resolution. In the slide-seq protocol, millions of barcoded beads, approximately 10 micrometers in diameter, are randomly deposited onto a surface. The precise location of every single bead is determined by imaging and sequencing its unique barcode prior to tissue application. A thin tissue section is then placed over this dense array, and released messenger RNA is captured by the underlying beads. This bead-based approach allows for transcript capture at a resolution that closely approaches that of individual cells. This enhanced resolution is crucial for resolving fine tissue structures and accurately mapping complex cellular interactions within heterogeneous environments. Imaging-based spatial transcriptomics methods provide extremely high spatial resolution by directly visualizing transcripts in situ. Multiplexed error-robust fluorescence in situ hybridization and sequential fluorescence in situ hybridization employ sophisticated combinatorial barcoding strategies. Target messenger RNAs are labeled with multiple short oligonucleotide probes. Sequential rounds of hybridization and imaging are then performed, where different fluorescent reporters bind specifically to these probes. The resulting sequence of fluorescent signals across multiple imaging cycles generates a unique binary barcode for each detected transcript. This unique barcode identifies the specific gene, allowing for the mapping of thousands of transcripts with single-molecule precision. This process occurs within the preserved cellular architecture, completely bypassing the need for sequencing library preparation and subsequent sequencing steps. Spatial transcriptomics data analysis requires the precise integration of the gene expression matrix with the corresponding high-resolution histological image, typically hematoxylin and staining or immunofluorescence. Fiducial markers embedded on the array surface facilitate the accurate computational alignment of the spatial coordinates with the tissue morphology. Specialized computational pipelines such as Seurat or SquidP are used to identify spatially variable genes whose expression patterns correlate significantly with their physical location. Clustering algorithms then group spots or cells that share similar expression profiles, effectively revealing distinct spatial domains within the tissue. Downstream analysis frequently focuses on mapping cell-cell communication networks by correlating the physical proximity of cells with the co-expression of known ligand-receptor pairs. Spatial transcriptomics is essential for dissecting the complexity of the tumor microenvironment, which profoundly influences cancer progression and therapeutic response. Spatial transcriptomics maps the precise spatial distribution of malignant cells, various immune infiltrates such as T cells, B cells, and macrophages, and stromal components including cancer-associated fibroblasts. Identifying spatially restricted gene signatures helps researchers accurately delineate tumor boundaries, invasion fronts, and areas of hypoxia. Mapping the precise location of immune checkpoints and immune cell exclusion zones provides critical mechanistic insights. These insights are vital for predicting patient response to immunotherapies and for developing highly specific, spatially targeted drug delivery strategies in clinical oncology. In neuroscience, spatial transcriptomics enables the high-resolution mapping of gene expression across highly structured tissues like the brain. It successfully resolves the molecular differences between distinct cortical layers, hippocampal subfields, and specific nuclei, thereby providing a comprehensive molecular atlas of neuronal circuitry. Spatial transcriptomics is extensively used to study neurodegenerative diseases by mapping the spatial relationship between pathological features such as amyloid plaques characteristic of Alzheimer's disease and the surrounding reactive glial cells. In developmental biology, spatial transcriptomics tracks dynamic changes in gene expression during embryogenesis and organogenesis. This capability reveals the spatial cues and morphogen gradients that actively drive cell fate decisions and dictate tissue patterning in developing organisms. The future trajectory of spatial transcriptomics involves integrating transcriptomic data with other spatial omics modalities. Spatial proteomics, utilizing techniques like imaging mass cytometry, maps protein abundance and post-translational modifications directly alongside ribonucleic acid measurements. Spatial metabolomics identifies the distribution of small molecules and metabolites within the tissue section. Current technological advancements are intensely focused on increasing throughput and achieving true subcellular resolution across the entire transcriptome. New methods such as in situ sequencing aim to sequence messenger RNA directly within the preserved tissue section. This promises whole transcriptome coverage at single-cell resolution, moving spatial transcriptomics closer to routine application in clinical diagnostics and personalized medicine.